Yeah, good afternoon, everybody. Thanks for joining. So I've asked Dr. Lucas Czech to give a bit of talk to us about his work on phylogenetic placement. So that's a method to help us study microbial communities and also other environmental DNA metabarcoding data as well, right?
Yeah.
So yeah, so he's currently at University of Copenhagen, and yeah, I hope we can learn a lot from you. And thank you for saying yes to the invitation. And please, yeah, you can start once you're ready, and we'll do some questions and answers afterwards. Thank you.
Thank you very much. Yeah, thank you very much for the invitation as well. This is quite fun. If there are any questions in between, also I don't mind if I don't know what the format usually is, but I don't mind if people want to ask questions in between. I also have an eye on the chat. If there's anything, or feel free to just open up your microphone if that is okay with the organizers, if that's how this is run. So from my side, that works.
Yeah, usually we do it at the end, but it's no problem if somebody wants to interrupt.
Okay, whatever works for you, just feel free to ask if you want. Otherwise, we can wait until the end. That sounds good. All right. Yeah, thank you very much. Welcome, everyone. So today I want to talk a little bit about phylogenetic placement, and I titled my talk Leveraging Phylogenetic Placement to Understand the Environmental Drivers of Microbial Community Composition. We'll go through most of these terms bit by bit, and I will also have a little, they will just come up in the talk. At each stage, we will look at different data sets and examples that I've prepared. And so just to give you a little bit of background, this was my PhD work back from 5 years ago by now. I've moved on a little bit to work on population genetics in the meantime, and now finding my way back at my position in Copenhagen now, which I started a few weeks ago, working again on environmental DNA and ancient DNA. And particularly, we are looking at ancient environmental sediments here. So I'm excited to kind of rejoin the topic of environmental DNA here in Copenhagen now, and I'm excited to see what will happen to that research in the next couple of years, and if I can pick up where I left all those years ago at my PhD. So today I'm mostly presenting the work from back then, and I will just give you a brief overview of how phylogenetic placement works, and then what the methods that I've developed during my PhD and what they can be used to. Let's have a little bit of a look at the motivation of all of that. I think you can see my slides changing. That should work for everyone now. All good?
Yes, yep. It does.
Yeah.
Perfect. Well, then let's go on. So motivation for that. Typically in metagenomic research, we have a couple of standard questions basically that we want to answer in the first place if we're looking at microbial samples, which is who lives there? That is the microbial composition of the samples, what type of bacteria or pieces of DNA from larger animals we would find in there, and how are these microbial communities composed of which taxa and in which abundances. And then we might want to start comparing samples to each other. How do they differ from different locations, different point in time, different weather conditions throughout the seasons maybe. There's a lot that we can do by comparing these samples. And then lastly, which environmental factors drive these differences? So is that seasonality, pH of the soil, or other factors that might change the composition of these microbial communities? And we'll have a look at all of these throughout the talk and methods that I've developed to help answer those. So one more recent example also that the group of Alexis Stamatakis, my PhD advisor, and I worked on during the pandemic was using phylogenetic placement to answer questions around the COVID pandemic, where we basically used it as a means of sorting out all those viral strains that were sequenced at the time and putting them on a backbone reference tree to just make sense from which regions we had how many reads and how they differ phylogenetically. Although it turned out that because these virus strains are so short with such a high mutation rate at the time that it was really hard to make phylogenetic sense of those. But we still tried using phylogenetic placement to that end as well. So the basic idea of why we want to use phylogenetic placement instead of something more simple like a sequence equality or sequence distance-based method such as BLAST or U-search, V-search is as follows. So on the left, we see a reference tree of the species that we're interested in, of the diversity basically that we expect in our samples. And what BLAST and other similarity-based algorithms do is they just give you the most closely related extant, so still living in the present in the database references that your data set from your microbial community might come from. However, phylogenetic placement gives an additional amount of information, kind of taking the phylogenetic history of these taxa into account that we have in our reference. So instead of just assigning basically to the tips of these species that exist in the database, we can also take the phylogenetic history and get placements, that's why it's called that, on the tree that describes the microbial community or the reference database that we're using to describe our microbial community. And so with that additional ancestral information, we can capture more distantly related communities or taxa. We can get a grasp of whether we might be missing something in the reference database, which might be overlooked by BLAST because it just gives you not so good scores, but you can't really see why. And with placement, you would kind of get that little bit of extra information that could tell you that you lack some diversity in your reference database to fully capture your samples. So with that method, some kind of broad range of applications that it has been used for in the past. So for example, one way is that it can be used is for data cleaning. That was a kind of the first paper there, the top line that I was involved in during my PhD, where we used it to have a look at forest soils in South American rainforests. And BLAST gave some really weird results there and not super good matches to the reference database, just because these soils were so hyper diverse in their microbial composition that nothing was even remotely similar, like everything was below 80% sequence similarity. And so phylogenetic placement was used there instead because it was able to work with this more distantly related data. Then other ideas or applications that have been used in the past, inference of new clades. So if you have a distant cousin of a clade that hasn't been described well in reference databases, phylogenetic placement can help to make sense and to put these in a phylogenetic context. It can be used for the estimation of ecological profiles, identification of low coverage genomes of viral strains, or for example, this virus data set that I mentioned before. And one data set that I've worked with before, clinical studies of microbial diseases. So this data set we'll have a look at later on. So this is human data, human microbial data that we will have a look at later as a really orderly data set that gives some really nice results there. All right, so with that motivation, now let's have a look at how phylogenetic placement works in the first place. Usually the typical idea is that we start with our multiple sequence alignment, or MSA for short, and a phylogenetic tree describing the evolutionary history of the species that we have in our reference. So we're always starting with some sort of reference that we already know, right? So this is maybe an important point to make. So in this set, just a toy example, we picked some larger animals there, and we have a sequence alignment and the corresponding tree on the right? So this is typical just normal using the normal phylogenetic placement, phylogenetic tree inference tools such as RAXML, IQ tree, fast tree, and whatnot to create this tree. And now what we have in our microbial samples is all those other samples, these short reads that we have from our metagenomic sample. And we want to make sense of that with respect to that multiple sequence alignment and reference tree that we have. That, of course, means that first we need to align those to the reference alignment that we have. There are specialized tools in the placement realm for that as well. And because that can be hard if you have so many references for whole genome sequencing, usually phylogenetic placement is constrained to some more conserved regions, marker regions, like 16S, 18S or something would be typically used there for the microbes or for the eukaryotes, just to constrain the analyses a little bit. Otherwise, you would need to make a whole genome alignment of all your short reads, and that is usually neither feasible nor recommendable because you lack a little bit of phylogenetic signal there as well, right? So now we have aligned all of our sequences to our reference alignment, and then the question for each of these reads that we get from our microbial sample is, where does that fit on our reference tree that we have? So this is the basic question that we want to answer here, right? And the pipeline that we use for that, which I just called the phylogenetic placement pipeline here, takes as input this reference tree and alignment that we have, plus all these query sequences, which is queries, is, if you hear me say that a lot now, is just the word that we give to these short reads that we have from our microbial samples. They're just called queries in the placement context, for short. I don't know who came up with that terminology, but it was what the papers that originally described these methods used, so I'm sticking with that as well. And we run one of the placement algorithms. There are a couple that I will mention later that basically place these sequences on the tree then, and we get this result that we see on the right there, the placements right on the tree. So this is basically, we can understand that, in other words, as a mapping of a single sequence to the branches of the tree. And technically speaking, this is an important distinction where there has been a little bit of confusion due to maybe not describing that well. Technically, it's a mapping of the sequence to all branches of the tree. So we evaluate every branch as a potential location and see how well it fits, how well that sequence would be fitting on that branch as if it was a branch branching out from that reference tree that we have there. And we compute a likelihood function, which is usually just the same that is used for at least the maximum likelihood methods in tree inference in the first place. So we're using the same likelihood model of how well that fits. And we describe that single sequence as if we pretend it was a new tip on the tree and compute the likelihood for that, right? So we have the query sequence Q there that is on a pendant branch inserted in between. We do not actually insert it as a new branch. Our tree, our reference tree always stays the same, but we pretend it is for the moment of the computation and then use that as a mapping of that sequence fits to that branch. But we don't grow the tree because otherwise with millions and millions of query sequences in our samples, we will just end up with a gigantic tree that no one could make sense of anyway. So we just use that for the mapping of sequences to the branch. And we do that for all, next slide here. No, not working. There we go. And we do that for each sequence for every branch of the tree. So that gives us a likelihood value of how likely that sequence is to be placed on that particular branch. And we can compute their ratio, which is called the likelihood weight ratio for each branch along the tree, which is basically just we take the likelihood of the sequence being placed on that particular branch divided by the sum across all branches. And so this is basically just a normalization that sums to 1 for all branches. And that can be interpreted in a way as a probability of the sequence to be placed on a branch. So if we visualize that, we would end up with these likelihood weight ratios, LWRs, for a single sequence looking like that. So we have a part on the tree on the bottom left that contains 0.23 and 0.57 of the probability mass in some interpretation of that sequence belonging to these two branches. So there's a high likelihood that this sequence belongs to that clade on the left of the tree, whereas the weights, these probabilities in the other branches are way lower. So with that, we can say, yep, there's a high confidence that this sequence belongs to this part of the tree. And we repeat that process for every sequence in our sample and just keep mapping these sequences to all the branches in the tree, right? So this is basically the data that we're getting out of the placement algorithm itself. And yeah, have that mapping of all sequences to all branches of the tree. For practical reasons, usually everything that is below a certain probability threshold is just discarded. If it's less than 1% or something, some user-defined threshold, we can just discard it because we want to keep our data a little bit more manageable. But technically, we have the full information of that placement. And we can summarize that as well. If we take the whole sample and sum up all those probability masses of each sequence being placed on each branch, we get a full distribution of how likely is it, of how much probability mass of all the sequences we get for each branch or each clade of the tree, right? And so there will be some pictures on some real world data in a little bit, but for now, just in the small scale example here, we can see that there's a lot going on, for example, on the lower two branches on the left there. And then I don't know if you can see my mouse cursor well enough, but like there's a little bit here going on as well and so on. So we get a nice summary of these, what I called edge masses, as a distribution of the whole sample of abundances basically, but in a probabilistic maximum likelihood phylogenetically informed framework of abundances on that tree, right? In more recent years, there have also been alignment-free k-mer-based placement methods. So here's a little timeline of how these methods came along, just to orient yourself a little bit. They don't use a phylogenetic likelihood framework, but instead alignment-free k-mer-based methods. So they have a little bit of some differences there, these newer methods, apples and rappas. Just to compare them a little bit, they don't have these uncertainty values that we calculate there. So what they give, let me walk you through that table. So we have the placement tools pplacer and RAXML EPA, which are the original ones from 2011 that use the maximum likelihood framework, and then EPA-NG, which is the more recent re-implementation of the RAXML EPA, also from the group where I did my PhD. All three of those, they use an alignment, they use maximum likelihood, they can give you these multiple potential locations per query sequence. So this is this multiple column there. And that also means they can quantify the uncertainty in your sample by having this placement distribution instead of just a single hit. And they can give you information on the branch length. I didn't mention that before, but basically we can say that the place sequences on the as well along the branches, so we get that information. Whereas these later three tools, the k-mer-based, they are faster, they lack certain capabilities just by the way that these methods work, right? So these are the different algorithms that we can use there. For the rest of the talk, I will focus on the top three, so the maximum likelihood framework, just because they have the most amount of information, of wealth of information in the data. The other ones might work for many analyses as well, but might fall short, in particular when it comes to the uncertainty where we need that. But I thought I'd give a shout out to those as well. Okay. Are there any questions so far, just for clarification, that people want to ask right away, in case there are any misunderstandings or any confusion? No. Okay. With that, then we can. Yeah. All good. Thank you. Just making sure that everyone is up to speed so far, right? So now let's have a look at some methods for analysis and visualization of these data that we get from the phylogenetic placement. And this dives into what I've done in my PhD and what other people have worked on to actually use that information for. So the basic idea of this phylogenetic placement as a distribution of sequences on a tree means that we have two interpretations that we can use for our interpretation. First, we can just say it's an assignment or mapping of sequences to branches. So each of our query sequences from the metagenomic samples, if we want, we could just look at each of them individually and say this sequence has been placed with that likelihood on that branch and so on, and use that. That is a typical use case if you want to be a little bit more specific and have a look at individual sequences. But typically what I've used on in the larger data sets, you want to have instead the second interpretation, where you just look at the whole distribution as a whole thing, right? So across and this is what the rest of this talk will also focus on. And typical types of analysis that we want to do in line with these open questions from the very beginning, from the motivation, is we want to look at a single sample. We want to compare samples to each other. Like a single sample would be, who is there, right? What are the abundances in the tree? Where are they high? Second question, relate samples to each other. How do they differ? And then the third question, relate these samples to environmental factors like the pH or temperature or something. Oh, 1 second. Let's have a look at the first, examining a single sample. So as I mentioned before, we can use this distribution along the tree as a means of assessing abundances in the tree. So this is from that first paper that I mentioned that I worked on, where we had a look at the rainforest in South America. And what we can see here is that the placements, these edge masses, this distribution summed up across all samples with up to 2.8 million sequences. It's not literal sequences because it's always these probabilities. So it's kind of fractional sequences in a sense, but it tells us pretty straight away that there is a high abundance in the clades on the lower left side of the tree with all the dark branches, right? And so we can kind of interpret that as a another more detailed way compared to the typical abundance charts that people would do with taxonomic assignments, where we would have these different clades and just abundance percentages, right? And we have the same information in the placement. We can annotate that even around the tree, as I did there, with the percentages annotated at the different clades of the tree. But we additionally get all that information within the phylogenetic context, where we get darker branches towards the inside of the tree, which would hint at some clade that is a cousin of a particular group of branches or something like that, that has a high abundance, right? So we get all that additional information. And we can use that if we wanted even to make a typical taxonomic assignment chart like this, like I think they're called Corona plots, where we would see this in a nested taxonomic context. So we can kind of remap our phylogeny onto a taxonomy and then use these per branch distribution values and interpret them as if they were taxonomic assignments, right? That works as well, but I prefer to stay with that visualization on the right, because that just gives a bit more detail even than that, while also still looking, I think that looks kind of cool to have that tree there. And just as a little bit of an excursion for practitioners of that, it might also be important to do a little bit of quality control on these things. So what we can have a look at here is, for example, the distribution of these likelihood weight ratios, where ideally we would want our tree to just have each sequence be placed with 100% certainty on one individual branch, right? That would be our ideal situation, where our sequences map perfectly to one branch on the tree only. And that would be that we see that the most likely, which is placement position, placement branch that we have for a given sequence, which is the dark color here, the first likelihood weight ratio of the first most likely placement location, would just be at 100%. So that right bar that is pretty high in that chart here already would just be at 100%, and the rest of the plot would be 0. In practice, we don't see that, of course, and so we get a little bit of a distribution like that, where the first most likely placement, the dark one, has more than, what is that, 20%, more than 20%, 25-ish% of all the sequences have a first most likely placement with greater than 95%. Wait, no, it's the other way around. 95, for everything that has 95%, there are 20% of the sequences that have that high of a confidence value, while there are fewer percentages in that histogram of darker sequences. So there's like this, these lower dark values tell you there's 5% that have something like a 50%-ish probability to be placed on one branch exactly, and so on. And then the two other greens are the second and third most likely, where we can just have a look at a little bit more detail of what other placement locations might be. So how fuzzy basically is our data. We can also visualize that in a more colorful way. So we would see here, for example, the first 5 different placement locations across all our metagenomic samples, and have a full distribution of that. On the left-hand side, we see a very fuzzy assignment with lots of uncertainty, because a lot of the mass in that plot of the accumulated likelihood weight ratio is in the yellow colors, which is the remainder, which is everything that is not even the 5th most likely placement for a given sequence, right? Whereas on the right-hand side, we have a lot of the dark colors. So that means a lot of the sequences in our metagenomic sample have a more or less high-ish certainty of their first most likely placement to be exactly where it is. So that is a little bit more certainty here. And this is also something where we can compare these maximum likelihood methods with the K-mer-based methods. So for example, the maximum likelihood method on the left would give a little bit more of a nuanced assignment to different branches. We see a lot of darker branches on the left-hand side there, and we see a lot of uncertainty in the yellow in the bottom part there. Whereas the K-mer-based, which are mostly like basically sequence identity-based in some sense, or K-mer identity-based methods, they are way quicker of just assigning a query sequence to that branch and that branch only, and give it 100% certainty, although a maximum likelihood framework would tell us, no, it's a little bit more nuanced than that, right? So this is kind of a little bit of a measure of speed versus accuracy there. If you have a lot of sequences or like a little bit of computational power, k-mer-based methods are faster, but they might miss a little bit of the nuance, whereas maximum likelihood is more expensive to compute, but gives you more detail and captures that uncertainty in your placement a little bit better. This can also be important if you have trees that capture a lot of diversity and you want to if you're not sure that your tree captures the diversity of your samples pretty well or something, then you might want to start with a maximum likelihood just to explore this nuance a little bit. Right, so this was exploring a single sample. We can also do that for multiple samples now. So for example, multiple humans, multiple locations in the forest or ocean, different points in time and so on. And typically we would also have metadata for each of these samples, right? We would have for human patients like a health status, we would have the pH value, the temperature of the water or something. And each of these samples would then, that we have from different locations or points in time, would then be modeled as one set of phylogenetic placements. All the sequences in the sample being placed on a tree. And then we get these, for example, 6 instances of placement results here of these distributions. And this is basically what we want to start comparing to each other then. Right, so we see here the dark branches in these 6 plots differ. They're in different parts of the tree. This is, of course, an example. So in reality, it would never be that clear, but this is just to give you an idea of what we're looking for. We have 6 different samples with abundances in different clades depending on where on some other properties of these samples, and this is kind of what we want to compare and make sense of and start seeing where do these differences in our samples come from. And that data set that I mentioned before that I played around with a lot in my PhD, because it has a really nice clear data correlation there, is from bacterial communities in women with bacterial vaginosis, which is a vaginal infection. And there were patients, I think 220 patients in total, that were where their microbial microbiome was sequenced and placed on a reference tree. And we can see that here for all the patients with these 2 branches or 2 clades on the left in very dark being the most prominent one in that data set, which are 2 different strains of lactobacillus that are indicative of a healthy vaginal microbiome, whereas a distribution of more diverse microbiome across the rest of the tree would indicate that this disease, this bacterial vaginosis, is more present in the patients, right? So one of the visualizations from the original paper of that data set looks like this, where we have on the right-hand side the taxonomic composition of these samples. And we see very clearly at the bottom, so the green and red columns, B and C, indicate the health status of the patients, red being severe disease and green being healthy. And we see that the healthy patients have these 2, in red and orange colors, these 2 clades of lactobacillus dominating the microbiome, whereas the diseased patients on the top of that right-hand side plot have a wide composition of taxonomically diverse microbiomes, right? And we can see that in a clustering tree on the left as well. So this is not a taxonomic tree, but it's a hierarchical clustering of the samples based on the placement though. So we basically used the placement on the trees to make a tree that is made via a clustering method. And we see that the top, the disease patients have a lot longer branches, so a lot more different sample composition in the abundances in their placement than the bottom part of the tree for the healthy patients, right? Another view, so this is the same tree, just rotated and colored, colored according to a clinical score called the Nugent score that describes the health status of these patients. And we see that everyone with a score of 0, meaning healthy, has really short branches on that clustering tree, meaning they have a very similar microbiome. And we clearly see these 2 different parts of the microbiome in the blue dots as well, the 2 different types of lactobacillus, whereas the diseased patients on the right have longer branches. That is a very different distribution in terms of their placement on their reference tree, right? So this is one thing that we can do to just distinguish samples and kind of ordinate samples with each other. With that in mind, though, there's a little caveat that is important to when working with that, because the metagenomic data that we're working with is inherently compositional. That means because of the typical process of how that works. We just take some genetic material, and whatever is there is either sequenced or amplified, which introduces additional biases, of course. We cannot really interpret the total number of reads in a sample, right? Like whether there are 1 million or 10 million reads in a metagenomic sample doesn't mean much. It could just mean more that we had more biological material to begin with or something like that, but it has no statistical value for us just as the number of reads, right? And so we need to keep that in mind. All that we can do is interpret these abundances relative to each other, right? And that like, is there more of this or more of that, right? And this is true for phylogenetic placement as well. And so what we can do to help with this composition, with the artifacts, statistical artifacts from this compositional type of data, is to transform that into a space that isn't compositional anymore. And there's a method called balances that have been adapted, to placements that does exactly that. So instead of looking at the per branch values that we did before, these edge percentages, this distribution on the tree, we now transform that into a per clade view. So we hence look at subtrees instead of single edges, right? And get a distribution, a transformation that takes care of the artifacts of this distributional composition. So we start with just quick walkthrough of that method, the edge masses that we have for one sample as before, and we look at a particular edge and then compare the abundances in the two subtrees induced by that edge. So on one side of the edge versus the other side of the edge, and we compute what's called a balance value. We don't need to go into the details. It's basically just the logarithm of the geometric means of these values for mathematical reasons, which basically gives us what's called a contrast between those. So for the dashed edge, what we get is a value that ranges from negative to positive values, telling us negative, there's more on geometric average, there's a higher abundance on the left-hand side in this R part of the tree. 0 means the trees are on geometric average the same in terms of their distribution of placement abundances, and positive values would tell us the right-hand side, the S subtree there, has a higher average abundance, right? And so if we repeat that process for every edge of the tree, then, and for each sample that we have in our data set, we have transformed everything from these absolute per edge assignments of masses onto branches to something that is relative to the clades. And that looks like this. So this is kind of, it's a transformation that also makes sense in a phylogenetic sense, to use that transformation phylogenetically. So what we see here is a path of red branches going down from the root of the tree towards that left-hand side, towards that lactobacillus clade, and then a couple of bluish, greenish branches across the rest of the tree. And this is basically what we expected from our samples, which means there's an anti-correlation. If we just compute correlation between these balances and our health status of the patients, we see this anti-correlation of red going to the lactobacillus, which means a high value in the health, in this health indicator, in the disease, like the more diseased a patient is, the fewer abundances we see in the path leading down to that lactobacillus clade. And we see a positive correlation for the other branches, where higher abundances in these bluish, greenish clades means that's correlated, positively correlated with the disease status, right? So we can use that transformation basically to make sense of the data in a little bit more informed way than, and capture information on all the clades instead of just per branch, right? So this also helps a little bit if you have reference trees that might miss certain bits of diversity because you capture the whole clade. You're not super reliant anymore on individual branches to be exactly correct. So that can help with that as well. And a different visualization from a different data set for that would be, for example, here, this is from oceanic data, where diatoms, like little microalgae, are negatively correlated in their abundances with salinity in the oceans, for example, just as a very little kind of second example of how that looks like, right? The more salt content there is, the fewer algae can survive in these environments. And so this is what we're seeing here in a reference context of that is suitable for that data set. And we can use these balances as well as an ordination method between samples. So we could compute a principal component analysis on these data, on these transformed values, and use that again to separate our samples. So the first HPC, which is the first principal component analysis of this edge-based transformation, would give us a separation into blue and red again, so into the disease status of the patient, up the blue ones into the 2 lactobacillus clades. We can visualize that on the tree as well, just to make sure that we're actually looking at the right type of information here. So with the first component, we kind of can map back these principal component values that we got from this transformed data back onto the tree. And we get dark branches towards that lactobacillus clade. So that means the first axis of differentiation between our data sets is along the path to that lactobacillus clade. And then the second principal component mapped back onto the tree would further differentiate between those 2 lactobacillus clades themselves, right? So it's basically this data, the 2 axes back on the tree, we can see to which clades they correspond to. Let's see, I'm a little bit running out of time, so I'll move a little bit more quickly through the next section. But this is a method that I came up with in my PhD that kind of tries to connect this more explicitly to the metadata now. So we start again with our transformed data that we have from these balances. And the method that I want to briefly mention here is called placement factorization. And the basic idea there is with these balance values that we computed, we indicate differences in abundances across clades, right? We split the tree into, for each edge, into the 2 parts, and we just have the differential in abundances across these clades, and we want to connect that to metadata that we have, temperature, pH value or something, and want to find edges or subtrees that are induced by these edges where the abundances in these clades change with metadata features. That's based on a method called phylofactorization that was originally developed for just phylogenetic trees. And the adaptation here is to use placement data instead of trees themselves. So we still use the tree, but instead of using the tree kind of as the input, we use the placement masses or their transformed balances as our input for the method. And we want to find edges or subtrees where something happens whenever some metadata feature changes. So for example, if we imagine a simple linear regression of these balance transformations with, let's say, some metadata here, the latitude of where an oceanic sample was taken from, we could imagine 2 different scenarios, imaginary edges, right, edge A and B, where depending on the latitude, whether we go from north to south here, the balance across edge doesn't change, right? So this computed value from the abundances that differentiates between is there more on this or that side of the edge in terms of abundances doesn't change, which means that edge, that branch of the tree is not important. Whereas on the right-hand side, with changing latitude, we also see a change in this balance, highly correlated. So that means that latitude is an important factor in this toy example here, where when we change the latitude, we get a predictable change in the balance that is in the differential abundance across this one edge that this plot belongs to, right? And we can use that as a means to split up our tree. This is why it's called factorization. We're basically factoring out parts of the tree that have stronger correlations with our metadata. So for example, in the first iteration, we would compute a fit of how well this correlation or this linear regression here that we saw here would be between each edge of the tree, each inner edge at least. And we would see here that edge 1, this darkest edge on the right-hand side between clade A, B, C, D, so the top part of the tree, and clade E, F, G, H, I, so the bottom part of the tree on the left. This edge 1 has the strongest linear regression fit between this balance description of the edges and the metadata that we're looking at. So we would just say, okay, this then just say this edge is kind of an important edge as our first factor that we want to take out of the tree, as this is important for the tree. This is an edge where abundances differ between these 2 clades that we're splitting here, depending on the metadata, right? And we can do that nested now for the remaining subtrees. So imagine now the tree is split into the two trees that are not connected anymore. So we only have a few inner edges left here. And we repeat the process, finding again a nested factor of the second most kind of, if you want, in a sense, highest predictable clade in the tree as well. And we split that one out as well, right? In that toy example, and we can continue for as many iterations as we want, finding more and more edges that are somehow interesting, that are somehow correlated with our metadata in terms of their abundances. And we can visualize that on a larger example as well. So again, using the bacterial vaginosis data set here as an example. So the first iteration would just again find our clade on the left here and would just have dark branches leading, so high correlation of the disease status with the balances on across our samples on that tree. And we would then pick, I forgot which edge exactly. I think it was like the middle one here or something. That was the highest one, but all of them pretty much have relatively high values of their of this kind of linear regression model that is used there, telling us that all the path down to that clade has a high correlation between abundances in that clade versus the rest of the tree in relation to like correlated with the health status of these patients, right? And in the second iteration, as before, we kind of see the differentiation between these two lactobacillus clades here as well. And we can use that as well as a means for ordinating our samples again. So the first factor again would split the red from the blue and kind of distribute all the blue ones along a line and keep all the red ones on the left-hand side of the plot. And then the second factor, the second iteration, would find the branch that kind of splits these other ones as well, right? And if we repeat that whole process for a couple more iterations, what we find is a tree like that, where the black edges mark the ones that were kind of factored out in each iteration. And then the colorful clades indicate the clades that were factored out with the description of which taxa are in these clades. And as you can see on the left-hand side there, in factor 1 and 5, we kind of split away these two different lactobacillus clades further. And then all the other ones that are colored around the tree are kind of the ones that are positively correlated. So this method, at least in the base form that I have until now, uses this linear regression fit as a means to describe that. So it can't distinguish between positive and negative correlation. It's just how well does the model fit? How well can we predict balances from the metadata? For future work, I want to use the kind of gradient or the direction of that fit as well. So kind of bring back something from the simple correlation to be able to tell that factor actually means that this particular metadata or this set of metadata columns that we have in our data has a positive impact on the abundances, whereas these other would have a negative impact and would reduce abundances in these clades. But for now, this is future work. At the moment, we just get information. This clade has predictability given the metadata in either direction, right? It could also be that your metadata has certain features that go one and others in the other direction. And because of the generalized linear model that is used in the background, that would still work because they would just kind of both work towards that sample's predictability in terms of its abundances, right? But that is a bit of future work. Yeah, and finally, before I'm coming to an end, the methods, all that I've shown today, also the ones, this hierarchical clustering and this HPCA that I mentioned, they were previous work that was done before my time, and all the other ones that I mentioned today that were my PhD work. They're all implemented in a tool that I called GAPPA. And if you're interested in a review that we wrote 2 years ago on this whole topic on the first decade of phylogenetic placement since these methods came out, that paper was actually what I think, as far as I'm aware, what got me onto the radar of Riel. So have a look at that, and feel free also to shoot me an email if you have any questions later on. I'm always happy to support and help phylogenetic placement to find an active community. Yeah, and with that, I'm coming to an end. I want to thank my PhD advisor, as well as all the institutions that I've worked at in the past. for supporting this work. Thank you very much, and time for your questions.
Thank you very much. And over to Riel to deal with the questions, but please raise your hand if you have a specific question.
I hope I wasn't talking too fast, by the way. I was looking at my watch a little bit on the meantime from now and then, and I didn't want to overdraw too much. I hope that was a manageable speed for everyone.
It was fine, thank you. Nicola, you want to ask something?
Hi, yes.
Yeah.
Thank you. Yeah, thanks for a really interesting talk, Lucas. The pace was great, actually, and very clear, because I think it's quite complicated sort of analyses on top of analyses. And
yes.
and you actually answered quite a few of my questions in the course of the talk, because initially I was wondering, you place those sequences sort of ancestrally, but what if there was a branch missing from the tree? But then you mentioned that clade-based placement, which I think would help with that.
Yep.
But then I was also wondering, to what extent is the whole method dependent on how accurate your tree is? And is there something you can do if there's controversies about particular resolutions in particular parts of the tree? How would you deal with that?
That's a very good question. I'm kind of answering both at the same time now, because the first one kind of relates to that as well. What happens if you have a tree that isn't describing your diversity well? So very good and very important questions. And there has been controversy about that, as you pointed out, in the past as well. So basically, let's answer first what happens, right? If you have missing diversity in your reference that is in your queries, but not in your reference, right? Your sequences would find some place to go. Ideally, you would either find that as some more basal placement of the sequence that would just be placed a little bit more up the tree in some cousin of a clade or something, or you might see it as a long branch. So because of this maximal Black-Lead framework, each sequence also is stored with the branch length that it would have if it had been properly inserted into the tree. So we have that information as well, and we can use the branch lengths there as well. Or you could see something like low likelihood weight ratio, so a very low probability density distribution for a particular sequence, for which there's a lack of representation in the reference, right? All these three things could happen, and it depends a little bit on the data, on the type of genomic region you're looking at, and many other things. And this is honestly an open question. So this is something also I think in the review, we mentioned that in the Outlook. It's an open question of how to quantify that, like how well is a sequence placed if it isn't placed properly, and how can we identify that, right? It's easy to see if it's properly placed. You would just get a high likelihood for the first most likely placement. That would just be 99% on that branch. Then you can be sure, yep, that's probably that, or at least very closely related. But any of the other reasons that I mentioned before could be indicators of something being wrong. And in that case, you would need to investigate where that is coming from. So one example, for instance, from this South American rainforest data set that I mentioned before, they found aquatic microbial parts in these forests. The running hypothesis is that they just rained down on the forest because it's rainforest. So it was kind of water vapor from the ocean that picked up a little bit of some microbes, or at least their DNA, and rained it down on the tropical soils, and then in the soil you found aquatic microbes, right? And so those couldn't be placed initially and were super weird to make sense of. And then the people that I worked on, the biologists that actually know more about how to do that properly, added references from marine microorganisms as well. And then these sequences kind of found a place in the tree and were properly placed on these branches as well. So this was kind of like an iterative refinement. Then on the other hand, though, what could also happen if you put too many references in your tree, like for example, you put different strains of the same species into your tree, and your query sequences would place well onto all of those, right? That would kind of dilute your analyses as well, because suddenly you see low probability, or at least distributed probability values for all of these branches. And they're all good fits because it's just different strains, and your phylogenetic resolution, your signal, isn't strong enough to differentiate between them. that could also happen if you put too much into your tree. So this is kind of like an art and a balance there. And for the controversy, I think the best way to avoid that is to just place it on different trees and see how much it actually matters and what significant differences you see. And if they're different, if you get vastly different results in terms of placement, then it might be worth investigating where that's coming from. If that's something missing in the database in your reference, or if the tree might not be well describing the sequence evolution. But this needs to be iterative, right? Like ideally we would want to have something like a gold standard tree for each community, each taxonomic group that people work on or something like that. But unfortunately that hasn't happened yet. And so for now, I think it's a little bit of playing around and seeing how well each of those works. Does that answer your question sufficiently?
Yes, definitely. Very interesting.
Okay.
Thank you.
Welcome.
Right.
Any other questions?
Any
Sorry.
other questions?
Can I ask a question?
Yeah, sure. Seth?
Thanks, Lucas. That was great. I struggle just a bit with the leap from this compositionality problem in metagenomic data, which is something I'm aware of. From the sort of leap from that to how this placement approach solves that problem. And I know you were sort of running out of time, and I suppose there's some technicalities there, but do you think you could try again? Sorry.
Sure. As for time, I'm happy to stay a bit longer. I don't know how quick you need to cut off this meeting, but I'm happy to stay a few minutes over for sure. And also we can catch up later on or privately if you want as well. But let me briefly go through. So you're referring to this slide and the one surrounding it, right?
Exactly.
So basically the placement itself doesn't solve the problem of compositionality, right? Like it's just, we place things on the edges and we have the same problem. Just the raw data kind of that we place, like these likelihood weight ratios or their sums across the sample, they're still compositional, right? You cannot change any of these values, of the blue values that we see here, without having to change the others, because they need to sum to 1. It's a probability distribution on the tree. And you cannot change one of those values without having to change some others so that the sum remains 1, right? So this is kind of this issue of compositionality in a nutshell there. And the way that this is solved via this edge balances or transformation that we do here is by computing this contrast value between the 2 clades of each edge. We are taking away this effect of, if we change one value, we have to change some others as well, right? In this green kind of transformation of the values that come out of this, we get a value for this dashed edge there, right? And that just gives us a contrast of, is there more on the left or more on the right? And we could put more on the left or put more on the right without running into issues where we would need to change that for other clades as well. This goes a little bit more in the math than I can remember after 5 years of not having worked on that. But somehow by using this, oh, there's someone drawing on the screen there. Somehow by using this log of the geometric means of the abundances, we get a nice kind of transformation of these values that allows us to make each edge, or this balance value for each edge, independent of the other ones, right? And so this is kind of the mathematical trick that transforms the data into a vectorized format that has, as you will notice, it has, what is it? 1 fewer degrees of freedom, no, 1 more degree of freedom than the original one. So by that you can change things without having to change the others. Is that more or less a little bit better as an explanation? If not, we can follow up in some other time if you want.
Yeah, so I think what I think I'm understanding is that you could repeat this for every sample,
Yes.
which has a community of bacteria. You get this edge mass of which characterizes the sort of distribution of that community in the tree with respect to the tree. And then you do this trick where you work out you do this, presumably you work out this balance value for every branch in the tree, every internal node in the tree.
Yes, for every branch and for every sample, yes.
Right. And then every sample will have a sort of vector of these edge balance values.
Exactly.
In order of all of the trees and all of the branches in the tree.
Exactly.
And then you can perform a PCA on those vectors to summarize.
Yes, that is exactly what's happening here.
Okay. Thanks.
Yeah, sorry. Like, I know that this topic is rather complicated. It took me ages to get my head around that when I was first reading about the phylofactorization that the method is based on. If I rushed there, sorry for that. I hope it makes a little bit more sense now. But yeah, basically we're just creating a vector per sample of these edge values, or a matrix for everything, basically, like one matrix that has samples and edges along its dimensions, and then we can compute PCA on that.
We have another question in the chat box. That you could do phylogenetic placement on 16S data. Does this mean that you can reanalyze 16S data for phylogenetic placement, or is different initial work needed? Can raw 16S data be used for the analysis?
Reanalyze 16. Raw, yes and no. Like, yeah, you would start with the raw data for sure. Like, you don't need to do any additional. If you have 16S of your samples, either by just amplification or having filtered out your whole genome sequences accordingly, you can start with that. The only step that you would need to do in addition is to create the alignment of those, right? And for that, I would refer to the review paper where we have a little section on that of the different tools that can be used that are specific to phylogenetic placement as well. In theory, you could use any aligner, but these specialized ones are specifically written for this type of data and are way faster because they just align the sequences to the given reference, but not to each other, because we don't need that information anyway. Placement takes every sequence as an individual sequence, and we don't resolve the phylogenetic relationship between the query sequences, so we also don't need to have them aligned to each other, right? And so we can just align all of them to the given reference. This is kind of the only pre-step that you would need to do, and then you can run, as I said here, with these alignments, you can run the placement algorithm. But in terms of the data, I can't think of a reason why it wouldn't work, like why your data collection or sequencing strategy would need to be different. Maybe that is something more for the biologists that have worked on that. I'm coming more from the computer science background, so sometimes I might lack a little bit of biological understanding of what's going on there. But as far as I'm aware, if you have 16S data and get it aligned and run the placement, that should work and should give you reasonable results, yes.
Yeah, I think so as well. Any further questions?
Well, I have a question. Kind of leading on this, on this slide now. For your reference alignment, is there like a golden rule of what's the maximum number of sequences to have? Because, like you said, too much can hamper things.
Yeah, too much of the same strain or species or something, I think. Yeah.
All right.
So in terms of, what makes sense there? So these K-mer-based methods that I mentioned before, they specifically in their publications point out how great they are for large reference trees in the hundreds of thousands of reference sequences. So you could basically just almost, maybe even by now, I haven't caught up with the literature in a bit, but maybe even now use full reference databases as your input, or at least the constraint to their 16S or 18S or whatever conserved region you're interested in. Maybe that works. Like, technically, these K-mer-based methods are capable of doing that. But the issue that I personally see with that is that it gets, like, it depends on your analyses, right? The methods, at least that I've worked on and that I know that the people that I've worked with are interested in, would make it really difficult to visualize a tree with 100,000 branches. But if you have some way of summarizing that information somehow, or if the downstream method that you want can make use of all of that wealth of reference, right? I don't see a reason why not. Maximum likelihood methods might be a bit too slow for that, so then you might need to resort to the faster K-mer-based methods and give up on the nice maximum likelihood resolved placements. But if you have a tree that big anyway, I don't think that matters too much. So long story short, I think it depends on what you want to do downstream. Whether you want to visualize the tree, then I would recommend keep it manageable so that you can print it on a piece of paper in your paper. if you have some other downstream steps that can work with larger trees, that should be fine as well.
Okay. All right. Cool. Thanks. Anybody else have a question, comment? Silence.
No, I think, thank you very much for that.
For sure. And if any of you have any further questions, as I said, feel free to shoot me an email.
Okay, so I would like just to use this opportunity to thank you very much for your time. It was really interesting, and I'm sure that I will get a few more students to have a look at this and to follow this. So it's a great talk explaining these methods and what's happening. So thank you to you for all your time, Lucas, and also for Riel for organizing this. So with that, I hope that you have a wonderful evening. Thank you.
Thank you. Thank you very much for the opportunity and the invitation.
Great. Thanks, Lucas. I will definitely send you an email at some point again.
Feel free. Yeah, sure. Absolutely.
Yeah, it was nice to have this personal contact, and then things can start from there. So thank you very much, and good evening to everybody. Right.
Thank you. Good evening.